We'll wait for others to join. Let's wait.
Yes, sir.
Okay. Good afternoon. We'll start this class. Let me share my slides. This is where we let off last time. All right, right. So this is where we ended up our class last time. The restriction enzymes are endonucleases which recognize, specific recognition sequences, right? Restriction endonucleases are also called as restriction enzymes, right? They come in many different kinds, right? You have type 1, type 2, type 3, type 4. Of this, it is type 2 that is most useful. Type 2 is most useful for our routine molecular biology techniques, right? Restriction enzymes also come in, different types, right? For example, there are some restriction enzyme which recognizes four base pairs. They randomly recognize combinations of this thing, four base pair. Some recognize six base pairs, and very rarely, some recognize eight base pairs. We gave an example of this four cutter, six cutter, and eight cutters. Let me see if I can go back to the slide. Right, there we go, right? So you have four base pair cutters like ALU1, MSP1, six base pair cutters like PBU2, KPN1. There are others like EcoR1, HINT3. These are all routinely used molecular biology restriction enzymes. Then you have eight cutters, which is not one. And then you have certain unusual restriction enzymes which can have, which can recognize unusual this thing combination. For example, in this case, I've shown an enzyme called MWO1, which recognizes GC followed by GC at the end, and then in between you can have any nucleotide. N means any one of the four, right? So it can recognize a broad range of this thing. So if you look at the frequency with which each of these are supposed to cut, it is given by a simple mathematical formula, which is, there are four base pairs, right? AT, TA, GC, and CG, right? Four bases to the power 4 or 6 or 8, depending on how many it recognizes. So if you do that simple mathematical calculation, then theoretically, uh, for every 256 base pairs, you are supposed to have four base pair cutter. A four base pair cutter is supposed to, recognize at least four times, approximately four times in a 1 kilo base pair fragment. If you look at a, six base pair cutter, it is supposed to do or cut every 4 kilo base pair, approximately every 4 kilo base pair. If you look at an 8 cutter, then the frequency of a restriction site, which is recognized by this 8 base pair cutter, theoretically is around 1 in every 65 kilo base pairs. Okay, but sequences are not distributed randomly. I told you classical examples of GC-rich genomes like Mycobacterium tuberculosis or AT-rich genomes like Plasmodium falciparum. You may have variation. You may have violation of this rule, but this is uh, to a large extent this holds good. Assuming that your ATGC is distributed randomly. Okay. Let's look at, last time I remember I said that New England Biolabs is one of those companies which have commercially made these restriction enzymes. This company itself was started in the 70s when the molecular biology revolution was just starting, right? Just starting, and they made a conscious decision to invest in both science and technology. And today it's a multi-billion-dollar company, right? It generates revenues, and that's why I keep telling you that, in your IPR class in the next semester, we will actually try to understand how to monetize the discoveries. Okay. So let's quickly look at this thing. One, I thought I should introduce to what these type 2 restriction enzymes are, and then how do you discover a new restriction enzyme. Suppose, I keep telling you this is just a theoretical fascination. Suppose, the fifth semester students went on some trip to a remote place, and they discover a new and pristine lake or a soil sample, and they discover a novel bacteria. That bacteria can have n number of enzymes, but one enzyme that will be of very, very interest to many people will be, does it have a new restriction enzyme? Assuming that you have discovered a new bacterial species, the question will be, does it have a restriction enzyme? Most have. So the question is, what kind of restriction enzyme is going to be there? If it is a restriction enzyme is there, how will you discover and how will you purify? So these are two videos, short videos that we will see, and then we'll go into the class. Okay. So this is the first of the video. So quickly, maybe you could note down, it talks about different these things. These are short videos, so I think.
Type 2 restriction enzymes.
Is the audio audible?
Yes, sir.
Okay.
Most molecular biology applications, such as gene cloning, DNA fragmentation, and analysis. They cleave DNA at fixed positions with respect to their recognition sequences. Over 3,500 type 2 enzymes have been characterized, recognizing over 350 different DNA sequences. Restriction enzymes are named for the microorganism from which they were originally purified. For example, Hindi3 was the third enzyme found in Haemophilus influenzae, serotype D. Type 2 restriction enzymes vary widely in size, amino acid sequence, domain, organization, and subunit composition, cofactor requirements, and modes of action. They are loosely grouped into subtypes based on their enzymatic properties. The four most important subtypes are type 2P, 2S, 2C, and 2T. Type 2P enzymes account for over 90% of the enzymes used in molecular biology. They recognize symmetric or palindromic sequences, 4 to 8 base pairs in length, and generally cleave within that sequence. The subunit composition of type 2P enzymes depends on the length of the enzyme's recognition sequence. Enzymes that recognize short, 4 base pair sequences act as monomers, comprising single protein chains, while enzymes that recognize longer, 6 to 8 base pair sequences typically act as homodimers, comprising 2 identical protein chains. Still other type 2P enzymes act as dimers of dimers, or homotetramers. These latter bind to and cleave 2 or more recognition sequences at once. Upon cleavage, some type 2P enzymes leave single-stranded overhangs, while others leave blunt ends. In contrast to type 2P enzymes, in which the amino acids that catalyze cleavage and those that recognize the DNA are integrated into a single protein domain, the larger type 2S enzymes, amino acids, are partitioned into 2 separate domains, linked by a short polypeptide connector. Due to this separation, the catalytic domain is positioned to one side of and several base pairs away from the sequence bound by the recognition domain, causing cleavage to be shifted to one side of the sequence. Type 2S enzymes generally bind to DNA's monomers and recognize asymmetric sequences, but cleave as dimers. In type 2C enzymes, restriction and modification activities are combined into a composite enzyme with 3 domains, one for cleavage, one for methylation, and a third for sequence recognition. Type 2C enzymes also cleave outside of the recognition sequences. Type 2T enzymes, in contrast to the previous 3 subtypes, use 2 different catalytic sites for cleavage, each specific for one particular DNA strand. Disruption of either catalytic site results in the creation of a DNA nicking enzyme that cleaves only one DNA strand. For more information about restriction endonucleases, visit www.nebrestrictionenzymes.com or go to Rebase, the restriction enzyme data.
Okay, so I would share the link of this. I don't think it is, this is publicly available, so it should not be an issue. You can go to New England Biolabs and you will be able to get videos, right? If you go to resources, you will get this category. You can click on videos. Okay, so you will be able to get these things. Let's quickly look at. So this is very important, type 2 restriction. This is a very common question. Many times it gets asked, So put some effort on, making notes of this. But this should be available, if you probably go and look at this catalog, you will get a beautiful description of what these things are. Okay.
So the restriction enzymes are found in bacteria or archaea, but not higher organisms. So you want to get some bacteria. So you can go anywhere.
So, you typically, most of these restriction enzymes are found in bacteria. They're not found in viruses. They're not found in yeast. Yeast has one, but that is not really useful. It's an intron-coded endonuclease. Okay, these are called inteins. These are not generally used. Okay, most restriction enzymes come from prokaryotes. Okay, the exception, I won't say exception, but other than bacteria, the others that carry restriction enzymes are blue-green algae, which are also called as cyanobacteria, right? So you have Anadina, Nostor. These are blue-green algae which have these restriction enzymes. Otherwise, restriction enzymes per se is found in bacterial species only, not in mammalians, not in viruses, not in any other organism. The moment you talk about restriction enzyme, that's why I told you, just assuming that you people went on a trip to a remote island and you scooped a soil sample or water sample and found a novel bacteria, the principal question will be, does it have any restriction enzyme? So restriction enzymes typically are found in bacterial species. Okay, that's why he talks that you need to get a bacteria on hand. Okay.
The restriction enzymes are found in bacteria or archaea, but not higher organisms. So you want to get some bacteria. So you can go anywhere. So, I mean, I've done them from, my brother's backyard, one of my best enzymes. I was out visiting and I just, took a water sample. I actually borrowed a little container from my sister-in-law. Took a water sample with some mud in it, brought it back, and try to pick different looking colonies. You pick the small one, the big one, the white one, the yellow one, and I get maybe 20 different organisms that look different on a plate. And then you just get pure culture, so you do basic microbiology. And then grow them up, break them open and say, mix them with DNA, and do they have an enzyme that cuts DNA and gives you specific pieces? If they do, it's a type 2 restriction enzyme. So then the fun begins. Just do a little purification, and then it's kind of like doing a puzzle, which I love. What we've done is we put a solution of our protein that we want to purify that still has some other things with it. So we want to separate our protein of interest away from the other stuff. So in this case, we put 5 mils of our sample of protein into this little tube. the pumps from the machine drive it through the tubing over onto the top of this column. It goes into the column, and we keep running a buffer solution continually through it. And the proteins actually migrate their way down through the column. So the solution is going to come down and then come over, and we're actually collecting fractions. This machine has a bunch of individual tubes for fractions, and it's going to collect a certain volume and then move to the next tube and then collect the same volume and keep going. Here we can see a trace of what's been happening. So the blue line is the UV absorbance. So it's going to, when the proteins come off, you'll get an increase in absorbance as they go past the detector. So there's basically nothing happening, and this is the protein we're after. Gave us a nice peak. And you don't see a whole lot else because this is already fairly pure. So this is actually looking really good. I'm happy with this. So I'll send this down to New York. They'll mix it with DNA, hopefully get it to form crystals, and then they can trace how the protein is actually structured. When you cut DNA, so you cut a few different DNAs or just one, they'll give you different size pieces. You can basically take some DNA, cut it, run a gel to separate things by size, and then look at it and say, oh, I have a different pattern of sizes. They're different enzymes. What we would like is to find new ones, so things that cut at a new site or cut even the same sequence a different way. So then you have more tools to use. This is a picture of an agarose gel. So you can see up here there are the wells or the holes in the gel, and I've put the DNA solution into these, which sit down into the gel, and then run a current from the top to the bottom. And the DNA migrates through the agarose. And it's kind of like people. The small pieces of DNA can work their way through the gel much faster than the big pieces of DNA. So you separate things based on size. So these are the largest pieces. They moved the shortest distance from here. And these are the smallest pieces. They've moved a significantly farther distance. So in this case, this particular one here is a DNA size standard. So these are known pieces of DNA that I know the sizes of. So that tells me where I am in the gel on this particular run. So it's the control, and it tells you I can compare this to my known piece, and this known piece is 1274 base pairs. So that's a little bit bigger. That's maybe 1280, 1290, 1300, something like that. The question I'm answering here is how much enzyme do I have? This is lambda DNA, which is one big intact piece, and it's cut with different amounts of enzyme. And we're looking at how much enzyme do I need to cut this into at every place where this enzyme recognizes the DNA. So it would be a complete digest. So these are complete digests. These are clearly incomplete. This is the highest concentration to lowest.
All right. Let's go back. Let's just type in Eco-R1. Okay, gives us okay, there we go. Gives you a lot of information, right? Remember last time I said one of the best ways to inactivate an enzyme is to heat it, right? So this one says, for example, this enzyme ACU1, okay, Is it heat inactivatable? Yes, it is heat inactivatable. The optimal temperature is 37 degrees centigrade. Okay, for almost all the enzymes, the storage temperature, the temperature at which the enzyme is stored, the optimal temperature is -20. But there are certain enzymes, although the optimal temperature is 37 while you do the reaction, they need to be stored at -80, right? So most of these enzymes need to be stored in deep freezer or in your -20. It also gives you, for example, for this enzyme, it says this is the recognition sequence, right? And gives you many different kinds of information, right? It starts from A, ends up with all the way to S. What I wanted to do was how do you define enzyme activity? Let us just take any one of them. Eco-Orbital is probably the best. I'm trying to see. I don't know why I'm not getting Eco-Orbital. These are type 2S, and we should be looking at type 2B restriction enzyme. Then give any let's quickly try this, and then we'll so each restriction enzyme, right, typically is tested. Before they ship it out to you, each restriction enzyme is tested on certain standard substrates. Okay, for some it is lambda DNA. Okay, so when you incubate with lambda DNA, depending on how many lambda DNA is about 50 kilobase pairs in length. Okay. So will it have a not one restriction site? Not one is supposed to be once in every 65 kilobase pair. There is not a very high chance that it will have a not one restriction site. On the other hand, will it have ECOR1 or a Hindi3 restriction site, which is supposed to happen every 4 kilobase pair? There is a high likelihood that it is going to have at least on an average about 10 different sites, right? It could be a little more, little less, but at least on an average, theoretically, it should have 10 different sites. Most restriction enzymes, are tested on lambda DNA. Some are tested on other specific kinds of plasmids. Whatever it is, when you buy restriction enzyme ECOR1, for example, they will actually tell you that this was tested on this particular substrate and gives you 100% activity. As I said, most of these enzymatic reactions happen at 37 degrees centigrade, right? You leave it for 1 hour, your DNA is going to be wherever it has a recognition sequence, it is going to go and act on those sites and it's going to cut the DNA. Okay. All right. Okay, one important thing, after you do your restriction digestion, one of the reasons you are doing restriction digestion, one purpose is to clone them into some kind of vector so that you can have clones of it. Okay, this is typically accompanied by doing a ligation reaction, right? So if you look at the ligase, okay, ligase that is typically used in the molecular biology lab comes from an enzyme called, from an organism called T4 phage, T4 bacteriophage. Okay, T4 bacteriophage has its own ligase. Commercially, this is the most commonly used ligase in almost all the molecular biology labs. Okay, although as I said, some people, not some people, but at least quite a majority have now changed to topoisomerase for routine cloning. Okay, you may still use. There are still people who would prefer to use ligase. If they use ligase, it is going to be T4 DNA ligase. Okay, E. coli has its own DNA ligase, right? When it replicates, when it does the lagging strand synthesis, or when there is a repair, this is the ligase that is going to be useful. So E. coli has its own DNA ligase. What is the difference between T4 DNA ligase and E. coli DNA ligase? It is the cofactor. Okay. For example, T4 DNA ligase uses ATP, uses the energy of the ATP. It hydrolyzes ATP and uses that phosphate to create this bond formation, okay, between the adjacent hydroxyl group and the phosphate group, which is highlighted by this green box. Okay. E. coli DNA ligase, on the other hand, uses NAD plus and converts into nicotinamide mononucleotide. Okay, uses the energy from that, okay, to convert this to make this phosphodiester bond. Okay, so only difference between T4 DNA ligase and E. coli DNA ligase is the cofactor that they use. Okay, E. coli DNA ligase is definitely a much larger enzyme compared to T4 DNA ligase. Okay, other than the fact that these are obtained from two different sources, the cofactors for these two ligase reactions are also different. Okay. So what is DNA ligation? DNA ligation is the process of joining two molecules. It could be the same molecule, in which case it is called as intramolecular reaction. It could be two different DNA molecules, right? It could be plasmid and a vector, in which case it is called as intermolecular reaction. What does it have? What happens is it creates the phosphodiester linkages. So you can actually imagine this is a nick in which it has a phosphate group and a hydroxyl group. And in this case, after you incubate them with the enzyme, the phosphate group and the hydroxyl group are getting reformed. And then this is the final product in which, the phosphodiester linkages have been regained. Okay, so that is how the ligation reaction works. Okay, this is a slide which I had shown to you long time ago when we started the class on this thing. So let's just say, this is a genomic DNA which has been treated with Eco-Orbital. It is going to generate hundreds and thousands of fragments, right? And this is your vector which has been cut with Eco-Orbital. Then you're trying to do ligation reaction, right? When you do a ligation reaction, you have intramolecular reaction. That means this fragment can ligate it back on itself. The vector can ligate back on themselves, or you can have this intermolecular reaction where the DNA vector picks up fragments of the genomic DNA. This is the most favored reaction. When we set up the ligation reaction, we set it up in such a way that you have large excess of the vector compared to the insert, so that you can have a higher chances of this getting ligated. In order to prevent this vector from religating on itself, that is when we use an enzyme called alkaline phosphatase. What does alkaline phosphatase do? A kinase transfers a phosphate group. A phosphatase removes the phosphate group. Okay, when you treat the vectors with alkaline phosphatase, remember the ligation reaction is between the phosphate group and the hydroxyl group. When you treat these vectors with alkaline phosphatase, this phosphate is lost. Okay, when this phosphate is lost, ligase cannot do its job. Okay, so typically when you treat these vectors, linearized vectors with alkaline phosphatase, this probability comes down. Okay, and the probability of these two joining becomes higher because this will carry the phosphate group, these will carry the hydroxyl groups, okay, and so the probability of the intermolecular reaction becomes much higher. Okay, is that clear? Can we move forward?
Yes, sir, we can.
Yes, sir.
Okay, so the behavior, so any idea what temperature do we do normal enzymatic reaction? What is the temperature at which in which we carry the enzymatic reaction?
So I'm just guessing, but is it 37 degrees
Yes, very
Celsius?
good. 37 degrees, right? We saw when we do restriction enzyme, most of them are at 37, but some of them need 25 degrees centigrade, right? Unfortunately, when we do ligation reactions, we end up doing ligation reactions at 4 degrees centigrade, okay? Any idea why we do ligation reaction at 4 degrees centigrade? Okay, so typically what happens is in our ligation reaction, we are having, so let us say this is your insert, this is your genomic DNA which has been cut with restriction enzyme, and these are the vectors, plasmid vectors. What you are seeing here is that there are regions of single-stranded DNA, right? When you incubate this at 37 degrees centigrade, the thermal vibration is so high that it actually, few more regions melt. So it is not perfectly, double-stranded this thing here. These termini are little bit apart. Okay, are you able to understand this? These termini fall apart a little bit, and so it becomes very difficult for the ligase to join these ends. So typically, in order to minimize thermal, vibrations, or these are also called as the ends are frayed. They move a little bit apart, they melt a little bit here. Okay, in order to minimize that, the ligation reactions are generally done at 4 degrees centigrade. Okay, and when you do any reaction at 4 degrees centigrade, it is not going to be that very optimal. That is one reason why when you do ligation reactions with T4 DNA ligase or E. coli DNA ligase, we generally end up doing it overnight. Okay, but unlike overnight reaction with T4 DNA ligase or E. coli DNA ligase, the ligation reaction that happens is topoisomerase happens at room temperature and in 5 minutes. So that is one reason why people have started shifting away from T4 DNA ligase into these topoisomerase mediated ligation. The only limitation to that is you need to buy it from an external vendor and who is going to charge. On the other hand, if you have T4 DNA ligase with you and you have plenty of time, you don't have to worry about those things. You can set up an overnight reaction when you leave college, and next day morning you can come and do the transformation. So it is no big deal, right? So it is, and T4 DNA ligase is fairly cheaper compared to those topoisomerase mediated kits. Okay. So the reason why they do ligation reaction 4 degrees simply because these termini, they behave differently at 37 degrees centigrade compared to 4 degrees centigrade. Is this clear or should I explain it?
Sir, could you just explain it once more?
Okay. So what you see here is a termini, okay, in which you have at the 5 prime, you have overhangs that are single-stranded, right? When you incubate this at 4 degrees centigrade, this is perfectly fine, okay. But when you incubate the same at 37 degrees centigrade, because of increased temperature, there is a lot more thermal vibration, and these ends, these free ends, they little bit stay apart. They stay apart. Not only this region, even few base pairs in these so-called which should ideally be double-stranded, which should be complementary base pairing should happen. It doesn't happen that easily. Okay, so typically what happens is it's like two ends that are staying apart. Okay, so the ligase finds it very difficult to join these ends. That is why ligation reactions are done at 4 degrees centigrade to minimize this thing, from moving out. Okay. So that is one reason why ligation is done at 4 degrees centigrade, and any enzymatic reaction which is done at a suboptimal temperature, it is going to take a long time for completion. All right. Okay. Can we move to the next slide?
Yes, sir.
Okay. So how can we make, so the ultimate aim of this ligation reaction is to maximize the intermolecular reactions, right? We don't want intramolecular reactions. We want intermolecular reactions, that is between the genomic DNA or cDNA. If you're making a library, it could be genomic DNA or cDNA and the plasmid vector. So we need to modify the end to make sure that the foreign DNA sequences are more ligatable, right? There are two different ways by which this can be done. One is called as directional cloning, which you can easily do. You will see some of the steps. It's nothing new. You already know it. Maybe we will see that in the form of a picture. Or treat the vector DNA with alkaline phosphatase, which I explained to you. So that typically prevents the recircularization of the vector DNA, and so you are essentially forcing the reaction to be more intermolecular reaction than intramolecular reaction. The plasmid cannot religate back on itself. Okay. So this is just an example. So let's say this is the foreign DNA, and you are cutting it with certain restriction enzymes, and it is going to generate, say, a termini like this, AATT. It could be GAATTC, which is the thing for recognition site for Eco-Orbital. Okay, you will cut with a similar restriction enzyme, the plasmid vector also, and by virtue of the fact that you will have a complementary base pair. Okay, these things will go and stick to this, and when you add DNA ligase, it essentially creates the phosphodiester bond, and you have perfectly circular plasmid DNA, which we can transform into E. coli. Okay, which I guess it's either you must have done E. coli transformation, or maybe you will be in the process of doing it in this semester. Okay. So ligation efficiency depends on the DNA ends in the reaction. The complementary or sticky ends, the ligation is very efficient. Only thing is we are going to do this at 4 degrees centigrade. The ligation process per se is very efficient. Okay. And annealing of the complementary overhangs essentially brings the 5' and the 3' hydroxyl to close proximity. As we saw, there are two types of ends that are possible. One is when you cut with restriction enzymes like Eco-Orbital or BAMX1 or Hindi3, it is going to give you what is called as sticky ends, right? When you do with certain restriction enzymes, they are going to create blunt ends. That means they cut right in the middle of the sequence on both the sides. So it's going to create what is called as blunt ends. The ligation of a blunt-ended DNA is a fairly inefficient process. It needs high concentrations of ligase and also the DNA. Okay. And typically what people generally do is to use certain reagents like polyethylene glycol 8000. Okay, these what they do is they do what is called as crowding. They crowd the space. Okay, and so essentially even in a dilute solution, the probability of vector and the DNA coming together increases because PEGs tends to occupy a lot more space. And by virtue of that, typically what happens is these are called as molecular crowding reagents. So they enhance the probability of a vector and the plasmid DNA and the foreign DNA coming together. Okay. and then you get dilute this, and then you do a transformation. Okay, but typically, when you buy ligase from any of the vendor, they will actually show you a protocol. They will actually give you a protocol that how to use if you're going to do a sticky end or complementary end ligation. But if you're going to do a blunt end ligation, they will also give you a typical protocol. And all you need to do is follow the manufacturer's protocol. By and large, most of these enzymes work fantastic. As long as you have stored all these reagents in the appropriate temperature. Okay. Most of them are, as I said, stored in -20, and very rarely you may need to store them in -8. Okay, you can do two other things. One is called, you can clone foreign DNA by adding what are called as linkers. Okay, now let's just say this is your foreign DNA. This is a perfectly blunt-ended DNA. Okay, this is a blunt-ended DNA. To this you can add what are called as linkers. Linkers, as you can see, are also short oligos or short oligonucleotide synthetic DNA, which are also blunt-ended. You can ligate them. Okay. Sometimes you can have more than one of them getting ligated. That is still okay. Don't have to worry about it. Okay. And these linkers will come based on the based on the restrictions. For example, in this case, it is called as an Eco-Orbital Linker. Okay. So this is an Eco-Orbital Linker. You can have a Hindi 3 linker. You can have a BAM-H1 linker. It comes in different restriction enzymes, based on different restriction enzymes. Okay. In this case, an Eco-Orbital linker is used. Linker is, as I said, nothing but something that has both the ends are blunt-ended. Okay. When you ligate it and when you cut with Eco-Orbital, it generates a complementary or sticky end. Okay. This you can cut your vector also with Eco-Orbital, and then you can ligate them. Right. When you ligate it, then you will be able to get this clone. Only thing is, we need to make sure that the Eco-Orbital doesn't cut right in the middle of your DNA fragment, right? You will be choosing a restriction enzyme. In this case, the example is Eco-Orbital. You will be choosing a linker which will not cut your DNA. No, you have cut your foreign DNA with some other restriction enzyme. Okay, and then you're going to add a linker which will not have any internal site. Okay, and then you will be ligating them, and then you will have a clone. Okay, one is called linker, the other is called as adapter. What is the difference between linker and adapter? What, as you can see here, an adapter has one end is blunted and the other end is a sticky end. You can actually see it as GATC, right? This is going to match with this. Okay, this G is going to match with this C, this G to this C, this G to this C, this C to this G, and this. So this is going to be, an adapter is something in which there is one end is blunt and the other end is sticky ends. Okay. So typically this is your foreign DNA, okay, which has a phosphate at the 5' and hydroxyl at the 3', and you will be ligating this. You will be ligating this, okay. When you ligate this, this is the product that you're going to get, okay. In this case, as you can see, the one of the 5' has a hydroxyl group, the 3' has a hydroxyl group, only one end as a phosphate group. As a result of this, typically what happens is this and this they ligate. Okay, this phosphate and this hydroxyl ligate. So what you end up is having 4 hydroxyl groups. Okay, and when you try to clone it, you're going to use a vector, and the vector will give you the phosphate backbone. Okay, to which this will go and ligate itself. Okay. The advantage of this is that you don't need to treat, this with any restriction enzymes. Unlike the previous slide, where you had to treat with the restriction enzyme after you ligate, in this case, you don't have to treat it. Okay, you simply do so this comes as a ready-made, right? This comes as a ready-made, ligated, and then you are ready to, ligate into the vector again, and then you can go for transformation. Of course, as I said, these are all technologies that were used in the 80s and maybe up to 90s. But nowadays, things have moved so much and so fast that if you were to be in a situation where you have found a, let's say a new bacterium for which you don't know, all you can do is to simply go back, okay. All you need to do is get a scoopful of that bacterial species. If you can culture it very well, unfortunately, more than 90% of the bacteria that we see in the soil or any of these samples, you probably can't culture them. As of now, the upper estimate is about 10% of the bacteria is only culturable. About 90% of them cannot be cultured. Okay, something that cannot be cultured, the technologies have matured to such an extent that you can still sequence them. Okay, all you need to do is you need to isolate extremely teeny tiny amounts of genomic DNA, and with all these linkers and adapters, people will be able to generate decent amount of sequencing data, and then you can go back and from that tiny amount of DNA, all you can do is start setting a PCR, and you will be able to amplify any gene of interest. Okay, so things have matured. These are all historical techniques by which people used to clone DNA. Another one was this terminal transferase, which we discussed in the previous class. Terminal transferase have this ability to add, nucleotides to the 3' end, and then you can, mix and match two different nucleotides, and then because of complementary base pairing, you can do the cloning part. Okay, this is all something that used to be very fashionable in the beginning days. Okay, so we are basically looking at the history of molecular biology. Okay, that's why I'm discussing all these things. Okay, one thing that we routinely do is PCR, right? PCR, when you do PCR with Taq polymerase, okay, Taq polymerase has this ability to add extra A. It invariably adds a nucleotide to the 3' end of the PCR product. Okay, it is non-template. It is not there in the template, but unfortunately, this enzyme adds A to it. Okay, so what people have been smart is to use vector which will have a 3' T overhang. For example, this enzyme called HPH1. When you cleave with HPH1, this is the sequence. Okay, the HPH1s, when you treat, it generates what is called as the T overhangs. Okay, now you can come with your PCR products that has a 3' A overhang, and when you incubate this and this, and when you add ligase, the ligase will seal the nick, and then you can clone the PCR. Initially, when people did PCR, people were very successful in doing the PCR. But unfortunately, they could not clone. They didn't understand why cloning is being such a problematic with PCR products until somebody realized that it is because of this process. Because of 3' A overhang, they are not able to clone the product. And eventually, this was also solved. Okay, so no longer an issue. Okay, the other is directional cloning, where typically you can use combination of enzymes, either single enzymes or double enzyme. People have started using, two different restriction enzymes, so that the probability of the vector coming back or the, insert itself ligating back is minimized, and you essentially are going to force, the entire this thing to be intermolecular reaction. Okay, so typically you are going to force this such a way that you will have vector and the insert getting ligated. Okay, so this is also called as directional or forced cloning. It is forced because you have prevented these non-productive, you can't have PST1 and Eco-Orbital. They can never ligate back. They can never ligate back simply because they are not complementary. Okay. On the other hand, if you have just Eco-Orbital or if just PST1, the vectors can ligate back on themselves. Okay. So by doing this, you essentially forced the cloning, right? So this also is very successful. Okay. When you do your PCR product, you can simply do one thing. You can have PCR primers in which you can incorporate known recognition sequences. As long as those recognition sequences are not in the PCR product, you can design PCR primers with inbuilt restriction sites. Okay. Only thing is we need to make sure that those sites are not here. Okay. As long as those sites are not here, for example, in this case, they've included AAGCTT, which is the recognition sequence for Hindi3, and CC80GG for KPN1 sequence. Okay, these are two different restriction enzymes, right? So after you do a PCR and when you digest, you are going to have, this is how it is going to look like, and it is very easy to clone this into any vector which has been digested with Hindi3 and KPN. So KPN1, and so this becomes ready for directional cloning. Okay, directional cloning, it's the most beautiful thing about directional cloning is that you are going to reduce your background. You're going to make sure that most of the recombinants will have your foreign gene of insert. Okay, 90% or greater than 90% probability that your foreign DNA will be ligated to the vector. Okay. Very rarely, it happens that you may see some clones that may look like a clone, but it will not have either foreign DNA or it is just incomplete plasmid DNA digestion, and that gets transformed. Okay, very rare. But okay, as I said, the last thing is to treat, with alkaline phosphatase. What does alkaline phosphatase do? So when you do a restriction digestion, okay, this is typically what happens. You have a phosphate at the 5' and a hydroxyl at the 3'. Okay, when you do ligase, this can go back on itself, right? This phosphate and this hydroxyl in the vector can ligate back. Similarly, this hydroxyl and this phosphate can ligate back. Okay, when you add alkaline phosphatase, typically what happens is it removes this phosphate. Okay, it removes this phosphate. Now, what happened is all of them are hydroxyls, right? So this reaction cannot happen. This reaction cannot happen. Okay, the only way you're going to get recombinant is because of this. Okay, you're going to have a 3' hydroxyl, 3' hydroxyl, 5' phosphate, and 5' phosphate, which will ligate to this. Okay, so this will ligate to this. You will still have a nick because remember this hydroxyl and this hydroxyl, they cannot join together. So you're going to have a nick. But this nick gets sealed once it is taken up by E. coli. Okay, so inside E. coli, this nick gets sealed and this plasmid starts replicating inside E. coli. Okay. So we will we'll stop in this thing. We'll start looking at how do we use this knowledge in creating genomic DNA library or cDNA library, as the case may be. It is, it is our decision whether we want to make a genomic DNA library or a plasmid DNA library. We'll have a discussion on this topic in tomorrow's class. Okay. Any questions on today's presentation? This slide I will share. Okay. Hopefully either today or tomorrow. Any questions so far?
No, sir, no questions.
Okay. 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 So if there are no questions, then we will meet tomorrow. All right.
Thank you, sir.
Okay.
Thank you, sir.